Hello, everyone. I'm Shannon Wyman, Director of Program Development at Keystone Symposia, and it's my pleasure to welcome you to today's ePanel event. Today, we will feature the awardees of the Michelson Prizes Next Generation Grants, an international prize that supports early career researchers who are applying disruptive concepts and inventive processes to advance human immunology, vaccine discovery, and immunotherapy research. The prize is a collaboration between the Michelson Medical Research Foundation and the Human Immunome Project to award $150,000 research grants annually to support young investigators from a wide range of disciplines. Here to share a few remarks about the prize is Dr. Michelson himself, founder and co-chair of Michelson Philanthropies and the Michelson Medical Research Foundation.
I'm delighted to join you today to honor the recipients of the 2024 Michelson Prizes. This year's recipients represent the sixth class of Michelson Laureates. These brilliant young scientists are pushing the frontiers of immunology and vaccine research. I created this prize to provide funding to individuals with incredible potential, but for whom the current system is stacked against them. The data shows that the most consequential breakthroughs come early in a scientist's career. Yet, there is virtually no funding to support that. We need to support transformative research in human immunology that accelerates vaccine and immunotherapeutic discoveries. Through this prize, we are betting on those who possess the knowledge, imagination, and perseverance needed to make these kinds of discoveries. The prize will also accelerate the career paths of visionaries who are fearless in tackling the unknown. We share this commitment with Hans and his team at the Human Vaccines Project, making them the perfect partner for this prize. Together, we support early career researchers with innovative approaches. Investments in science and medical research are a down payment on our future. They will lead to more breakthroughs, save more lives, and reduce human suffering. Thank you all for joining us today. Together with my wife, Alia, we extend our congratulations to the winners.
Thank you, Dr. Michelson. And next, we have Dr. Hans Kierstead, CEO of the Human Immunome Project, to comment on their partnership in sponsoring the prize.
It's wonderful to be here. I'm Dr. Hans Kierstead, CEO of the Human Immunome Project. As a former researcher, I know the power of catalytic funding for scientific discovery in individual careers. And as a biotech entrepreneur, I appreciate the importance and the difficulty of developing new vaccines, drugs, or immunotherapies. And as a nonprofit executive, I understand the urgency of delivering impact for patients worldwide. These experiences, for me, underscore the significance of the Michelson Prizes, Next Generation Grants, for both the winners and the beneficiaries of their work. So I'm excited to be with you to celebrate these three outstanding young researchers. But first, I'd like to thank the Michelson Medical Research Foundation and Dr. Michelson specifically for their commitment to supporting early career scientists and for their partnership on the Next Generation Grants. We at the Human Immunome Project are honored to partner with the group on this effort. The prizes are uniquely positioned to advance the field of immunology by supporting the sector's emerging leaders, and we need look no further than this year's recipients to understand its immense impact. To this year's winners, Dr. Ding, Dr. Otero, and Dr. Schaefer-Babajew, a massive congratulations. This year, the prizes became much more competitive. Your proposals stood out for their novelty, innovation, and most importantly, potential for impact. We're thrilled to support your work, to watch your progress, and see the outcomes of your award-winning research. As the sixth class of the Michelson Laureates, you now join an esteemed group of researchers that are deepening our understanding of immunity and helping us chart a new future for global health. We're proud to play a small role in your transformative efforts. So thank you and a heartfelt congratulations.
Thank you, Dr. Kierstead. And finally, we have a few words from Congressional Representative Ted Lieu of the state of California as to the importance of funding these innovative projects and the scientists behind them.
I'm Congressman Ted Lieu, and I represent California's 36th Congressional District. I want to thank Dr. Gary Michelson and the Michelson Medical Research Foundation for your outstanding work in making the world a better place. I also want to thank Dr. Michelson and the foundation for being a founding partner of the California Institute for Immunology and Immunotherapy, which will be located right in the 36th Congressional District at the UCLA Research Park. Now it is my honor to celebrate the winners of the Michelson Prizes Next Generation Grants. This year's winners, Dennis Schaefer-Babajew, Claire Otero, and Siyuan Ding, are doing exciting work that will advance our understanding of antibody feedback, vaccine development for CMV, and enteric viruses. The Michelson Prizes, done in partnership with the Human Immunome Project, are awarded to highly innovative and impactful research that has the potential to be applied to many diseases. The prize is intended to encourage, support, and help advance the pursuits of researchers early in their promising careers. The Next Generation Grants can help support researchers at a critical time. Traditional funding sources often overlook forward-thinking, innovative ideas from scientists who have just begun their careers. Greater investment in biomedical research from all sectors, philanthropy, business, and government is needed. Congratulations again to the 3 winners, and thank you, Dr. Michelson, and to the Foundation for your continued commitment to our future.
And now it's my pleasure to introduce this year's awardees, who are working to better understand immune responses against a wide range of viral and bacterial pathogens in an effort to better design protective vaccines. After I introduce each awardee, we will show a 10 to 15-minute presentation about their work. Once all of our awardees have presented, we will have a collective group Q&A. You may submit your questions at any time into the Q&A box and vote on others' questions that you would also like to hear answered. Please note if your question is directed to a specific speaker in the text of your question. And now it's my pleasure to welcome our first awardee, Dr. Dennis Schaefer-Babajew, a clinical scholar in the Laboratory of Molecular Immunology at Rockefeller University. Dr. Schaefer-Babajew studies how prior exposure to vaccines or infections influences subsequent immune responses, in some cases interfering with protection. His work has implications for HIV, influenza, SARS-CoV-2, and many other pathogens that we are repeatedly exposed to, and whether booster vaccinations may help or hurt us in terms of protection. Thank you for joining us today, Dr. Babajew. And Dr. Babajew's proposal is titled Antibody Feedback Regulation of Adaptive Immunity to Cognate Antigen.
My name is Dennis Schaefer-Babajew, and I'm an instructor in clinical investigation here at the Rockefeller University, and I study B-cell and antibody immunity. I was awarded the 2023 Michelson Award for my project working on a concept called antibody feedback, which is in other words the question how antibodies from a prior antigen encounter affect their immune reaction to subsequent encounters with the same pathogen. When we talk about secondary or repeat antigen encounters, we don't have to just consider infections, but in fact also a lot of vaccination regimes. Specifically, these are important, like repeat vaccinations, when we're talking about rapidly evolving, fast mutating pathogens such as influenza and HIV. And in this case, it's been long thought that repeated booster vaccination doses are necessary to shepherd the development of a couple of very precious and very peculiar B cells over time. We actually think, though, that the concept of antibody feedback and how the pre-existing antibodies from an earlier immunization then subsequently affect the second and the third and so on, that will have an effect that may be actually detrimental to this goal. And really proving that and showing how that potentially, hopefully, can be circumvented is, of course, critical if we ever want to create a successful HIV vaccine. To answer the fundamental question of antibody feedback in a level of detail and rigor that we are hoping to do, we need to use a lot of state-of-the-art technologies, which are quite expensive. So the financial support that the Michelson Prize affords us will help a long way to bring this from just an abstract idea to an actual discovery. But in addition to that, winning such a prestigious award will hopefully also raise more awareness for this particular question that we're investigating and bring the concept of antibody feedback more into the collective consciousness of the scientific community. And who knows, maybe someone is going to pick up the pipette and start asking the next question in this realm. I'm Dennis Schaefer-Babajew, and it is a great pleasure and honor for me to be here today and to be able to tell you a little bit about the project for which I was awarded one of the 2023 Michelson Prizes. So what I want to focus on today is to first introduce you to the concept of antibody feedback in a few words, and also give you a little glimpse into the history of the research on this question, then go over some of our published data that I hope will illustrate some of the major aspects of this mechanism to you, and lastly tell you about our thoughts on the potential ramifications of this mechanism and how we're currently working to address the impacts of antibody feedback on longitudinal immune responses. So, when encountering an antigen for the first time, say through an infection with a particular virus, we usually mount a B-cell response that leads to the production of antibodies, the levels of which rise at a certain speed and up to a certain level, and then these antibodies slowly decay after the threat hopefully subsides. Now, it is well known that a second encounter with that same antigen leads to an immune response that is different from the first one, and that it's usually more rapid and also more vigorous. While this general observation is essential, I would even call it an axiomatic feature of adaptive immunity. If and how these antibodies generated during the first encounter shape the subsequent response in some sort of feedback manner is a question that has actually been puzzling researchers for well over 100 years. There is a rich body of literature on this question, a few studies of which I try to highlight here. However, despite the large number of studies, including some of the more recent ones listed here, the whole mechanism seemed to be fairly unclear because in some cases, pre-existing antibodies were found to have a negative feedback effect, while in other cases they seemed to have an enhancing effect. And it was all somewhat context dependent. And so except for the general, very vague notion that pre-existing antibodies probably have some ill-defined feedback role, exactly how they do so remained entirely unclear. To try to shed some light on this and to better understand the role of pre-existing antibodies in shaping an immune response, we took advantage of a pretty unique opportunity of a phase 1 study here at Rockefeller. And in that study, 2 monoclonal antibodies, C135 and C144, had been tested in healthy volunteers for safety and tolerability, as is customary for a phase 1 trial. And they were safe and tolerable, and after the trial, most participants also went on to get one of the 2 new mRNA vaccines against COVID. So, knowing this, we asked them to come back to the clinic for blood draws after their first and second vaccine dose, so we could study their immune response to the vaccine and compare it to that of control individuals who were also vaccinated with the same regime, but had not received the antibody pre-infusion. And so, using this super clean and very controlled setup, we then looked at, for one, the circulating antibody levels by ELISA and neutralization and other serological methods, but also at their immunization-induced memory B cells in these people using single-cell techniques. And we supplemented this data with some experiments in mice, where we tried to see if our findings from the humans, which I'll tell you about in a second, could also be extended to germinal centers in situ. What we found, and I'm summarizing really a lot of data here in a very few words, so bear with me, is as follows. The composition and the magnitude of circulating antibodies in the pre-infused individuals was largely unperturbed, and which this suggested to us that the plasma cell compartment is relatively unaffected by this pre-infused antibody. In contrast, however, the memory compartment in these individuals was really dramatically altered. We found that these individuals had substantially more memory B cells of a presumably germinal center independent origin, and these were predominantly of a low affinity and also targeting alternate epitopes, specifically ones that were not targeted by the infused antibodies. And when we then looked in the germinal centers of mice, we found pretty much similar trends. So overall, lower affinity cells participating, and these cells were also binding to different epitopes. And as I already mentioned, this is all published work, and you can find that here. And reassuringly, I also want to add a whole host of other groups have since shown similar findings, which is always nice to see. Now, based on our study, we propose a dual mechanism model for how we believe antibody feedback operates. When there are no pre-existing antibodies, B cells, which you see here in the different colored circles, encounter antigen mostly as a soluble, low valency particle of some sort. And I'm illustrating the different B cell specificities here with different colors, and the cells with higher affinity with more shading, so like the more saturated circles. So in this setting of no antibody feedback, only cells with the highest affinity will be selected and recruited into the reaction, and they will eventually, through the germinal center and the alternative pathway, end up in the memory pool. In contrast to this, when there is pre-existing antibodies, say of this red variety here, coming from these red cells, the antibodies will now bind to their particular red epitopes and trap that antigen on follicular dendritic cell networks, thereby creating sort of a carpet of now multivalent antigen. And so what this does is that on the one hand, this high avidity setting of these immune complexes will allow cells of even subpar affinities to participate in the reaction. So these are all the pale circles here. And at the same time, red cells will be relatively excluded from the reaction because, again, these red antibodies, the pre-existing antibodies, are masking their epitopes. And so that's why you end up with a low affinity pool of cells that is devoid of this particular specificity. Overall, we believe this will lead to a diversification of the immune response. So these two mechanisms of avidity and also of epitope masking. And this, in some settings can be a very good thing, like in the case of COVID. But we believe in other cases, this may not be so favorable. And I'll explain what I mean by that in just a second. Now, at this point, I want to take a step back to reflect on why we sometimes even have to vaccinate multiple times for the same pathogen. The first one is the first reason. That's just to really increase the magnitude of the response, just like I showed you in the first slide. But in the case of certain highly mutating problem pathogens, just to name two, influenza and HIV, for example, it has also long been thought that sequential vaccination could be a good strategy to select rare and peculiar B cell clones with a particular immunogen, and then expand and affinity mature these desirable, let's call them B cells, over multiple rounds of immunization, so like here, to eventually elicit so-called broadly neutralizing antibodies, which are high affinity cell-derived antibodies to particular very vulnerable epitopes. And these are thought to be essentially crucial for protection. And I tried to illustrate this process the way it was envisioned here, where there's an enrichment over time in red cells and they get better and better. That means darker and darker. Now, while the first goal here, the boosting, clearly works, and you can find this in any immunology textbook of your liking, these latter two goals seem to not be so easily achievable. And in fact, more than a decade of failed attempts to create an HIV vaccination regime perfectly illustrates this point. We think this has, at least in part, to do with antibody feedback, because taking what I showed you today into account, what we believe probably happens in such a sequential response is that the antibodies that one generates following a first immunization, say again of this red desirable, let's say, variety, will then, in a subsequent vaccination, divert the response to other epitopes than their own, the ones that they essentially block through epitope masking. And at the same time, by forming immune complexes, they will create essentially less of a selection threshold for even low-quality B cells to be incorporated into the reaction. And the net effect of all of this will essentially be a diverse but low-quality response to all sorts of irrelevant off-target epitopes. Essentially, in a way, like shooting yourself in the foot through these multiple vaccinations. So we started this project asking the simple question whether antibody feedback is real. And I do hope, even in that short amount of time today, that I could convince you that it is in fact real, because we could observe it and so did other people. I hope I was also able to show you that through our work and that of other groups, we now have at least some preliminary answers to the question how, by which mechanisms that is, pre-existing antibodies exert this feedback. But I want to point out that this is not a solved question, as many mechanistic details still need to be resolved. However, while we now have a theoretical framework that implicates antibodies as one of the reasons for why many sequential immunization strategies have failed to deliver on their promise, as this cartoon up here shows, We still need to actually formally prove this in a bona fide loss of function type of experimental manner, and this is something we're currently heavily working on. And last, we will then have to see and test experimentally whether removing this antibody feedback from the equation will finally allow for the design of a successful HIV vaccine strategy. And with this, I want to really thank the people who made this work possible. And today, time doesn't really permit me to speak to all of their contributions individually, but I hope you can see from this long list of people that this whole project that I was presenting kind of in an abbreviated form in only 10 minutes today has truly been a team effort. And with all the experiments that we continue doing, it also continues to be such a team effort. I also want to take this opportunity to thank the Michelson Foundation and the selection committee for recognizing the work with this wonderful prize and also for supporting me going forward with this project. And finally, I want to take the time to thank the organizers of today's event for giving me the opportunity to present this work to you and all of you for your attention. Thank you.
Thank you, Dennis. Our next awardee is Dr. Claire Otero, a postdoctoral associate in pediatrics at Weill Cornell Medical College. Dr. Otero is working on developing a vaccine against cytomegalovirus, or CMV, which is typically asymptomatic or causes mild symptoms in adults. However, when passed from mother to infant, it can cause severe disease that can result in loss of hearing, vision, or brain damage. Her approach is targeted against the viral proteins that bind to and remove host antibodies from circulation as a means to evade host immune responses. By blocking this viral defense mechanism, she hopes to free the immune system to do its job in controlling the infection. Her proposal is titled Viral FC Receptors as Vaccine Antigens for Prevention of Congenital CMV. Thanks for joining us today, Claire.
My name is Claire Otero, and I'm a postdoctoral associate in the Department of Pediatrics at Weill Cornell Medicine. I received a 2023 Michelson Prize for my work in vaccine development for prevention of congenital cytomegalovirus, or CMV. I was interested in studying virology when I started graduate school, and my Ph.D. advisor, Dr. Sally Permar, introduced me to CMV immunology. Over the course of my PhD studies, I have developed a passion for CMV research, primarily because congenital CMV is so devastating for some families. But as an infectious disease, it should be preventable. Vaccine development work has focused heavily on developing antibodies that can neutralize CMV, but recent studies have suggested that other types of antibody functions are more important for preventing vertical CMV transmission. These functions involve interactions with immune cells to signal effector functions like killing of infected cells through cytotoxic granules or phagocytosis. However, CMV encodes multiple proteins called viral Fc gamma receptors that help it to evade these exact antibody functions. My research project focuses on developing vaccines that target these immune evasion proteins with the goal of improving these critical antibody effector functions. This funding enables new aspects of my preclinical investigation of CMV immune evasion proteins as potential vaccine antigens, including the development of monoclonal antibodies that will be useful as research tools and potentially even as therapeutic agents themselves. I hope that this Michelson Prize might also bring a little bit of awareness about congenital CMV and its impact. Awareness of congenital CMV is low relative to its impact, and it's important for pregnant people or people trying to get pregnant to be aware of CMV as a risk to their infant so they can take steps to protect themselves. Hi, everyone. My name is Claire Otero, and I'm a postdoc in the lab of Dr. Sally Permar at Weill Cornell Medicine. And I'll be talking today about our Michelson Prize funded project, Viral Fc Receptors as Vaccine Antigens for Prevention of Congenital CMV. So CMV, or cytomegalovirus, is the most common congenital infection and cause of neurologic growth defects, most commonly hearing loss, with about 1 in 200 babies born with congenital CMV in the United States, although incidence does tend to be higher in developing countries. But we know from epidemiology studies that placental transmission occurs in about 30 to 40% of seronegative women experiencing a primary infection during pregnancy, but this drops drastically to only 1 to 2% in seropositive women with a secondary infection or viral reactivation during pregnancy. And this suggests that the experience of the maternal immune system can protect against vertical CMV transmission, and thus that vaccination is a promising strategy for prevention of congenital CMV. However, this has been a priority in vaccine development for decades, but we still don't have a licensed CMV vaccine. And this may be in part because vaccines to date have largely focused on eliciting neutralizing antibodies. But there's this growing body of evidence to suggest that neutralization alone is not sufficient for prevention of CMV acquisition, viremia, or vertical transmission. But a few recent studies from a former graduate student in the lab have suggested that Fc-mediated effector functions are more important for protection from vertical CMV transmission. So these include functions like antibody-dependent cellular phagocytosis, or ADCP, and cytotoxicity, or ADCC. But interestingly, CMV encodes multiple proteins that we call viral Fc gamma receptors that function to invade these exact immune functions. And they do so through a couple of distinct mechanisms. So first, bipolar bridging is where the IgG will bind at its antigen binding region to the CMV antigen that it's specific for. And then viral Fc receptors in the vicinity at the surface of the cell or on the surface of the virion can come in and bind to the Fc portion of that IgG. And in some cases, this is sufficient to prevent interaction with Fc receptors on host effector cells. But these can also facilitate internalization of this entire immune complex, which more completely removes that antibody from detection from those effector cells. So what we know is that ADCP, ADCC, these Fc-mediated effector functions seem to be important for prevention of vertical CMV transmission, but CMV encodes these proteins that help it to evade those functions. And so our idea is, can we improve these Fc-mediated immune functions by targeting the viral Fc receptors by vaccination? And an important thing to note here as well is that exceeding infection-elicited immunity has been a primary goal of CMV vaccine development, but vaccines to date have fallen short. And so we're excited about the idea of targeting an immune evasion mechanism because it may help us to achieve this longstanding goal. So in order to do sort of a proof of concept study, we are utilizing a rabbit immunogenicity, as rabbits have been shown to be a good model system to evaluate these Fc-mediated immune functions. And so we have started by modeling the most efficacious CMV vaccine that has been trialed thus far, which is a glycoprotein B subunit protein vaccine adjuvanted with a squalene emulsion adjuvant. And then we use that as a baseline and then add in each of the individual viral Fc receptors to evaluate its impact in the immune functions that result. And just to give a little bit of context, GB, or glycoprotein B, is the primary protein that's responsible for membrane fusion during viral entry. So this has been a very popular target of vaccine trials and is a very important protein in the viral entry process. So you can understand why it has been such a protein of interest in this space. So for our vaccine schedule, we gave these vaccines for each of our 4 groups in a 3-dose series, each 4 weeks apart, with blood draws every 2 weeks. And we took this out 16 weeks following the initial immunization. And What we're expecting here is really that responses related to neutralization will not differ, but those Fc-mediated effector functions will improve. And just to get to the punchline, when we look at CD64 or FCR1 activation using an IL-2 reporter cell assay, we find that inclusion of GP34 significantly improves CD64 activation over the GB-only vaccine group. And there was a marginal improvement by GP68 and GP95, but this didn't reach statistical significance by any means. And this pattern is mirrored in the ADCC responses over the full time course with the GB plus GP34 vaccine, resulting in higher ADCP responses compared to the GB only group, starting at week 6 and continuing through the remainder of the vaccine schedule. So, taking a step back, we can see that this impact on the CD64 activation and downstream ADCP function is not due to differences in binding to GB. As when we measure these responses by ELISA, these are remarkably similar across all 4 groups. And similarly, when we look at FCR binding to the GB-specific IgG, this is also remarkably similar among the vaccine groups and follows a similar pattern in terms of binding to, or the total IgG that's binding to GB. So just to wrap up sort of our preliminary results so far, we've found that CD64 activation and functional ADCP responses are greater in rabbits immunized with GB and GP34 over GB alone. And this is not a result of the GB-specific IgG responses, as those are very similar between the 4 groups, and also not a result of differences in FCR binding to that GB-specific IgG, as this is also very similar between all of our groups. So what we've done so far is really just one piece of our overall hypothesis. And so we have a lot of work to do still with our next steps in evaluating the responses against the viral Fc receptors, which is a little bit more complicated because these proteins function to bind to immunoglobulins. So we need to utilize Fab-only approaches to avoid the non-specific interactions with the Fc portion of the IgG. And then we also want to do some work to evaluate what the mechanism of action here is, because these Fc-mediated antibody effector functions could be improved through, our primary hypothesis is that they're blocking that immune evasion function, but antibodies against the viral Fc receptors could also mediate these functions themselves. So we want to parse that out a little bit further. And then, of course, we also are working to evaluate the ADCC responses, which is something that I'm working on currently. So to talk a little bit about the Michelson Prize and what this is funding, we have some exciting proof of concept data, but we still have some work to do to optimize this vaccine formulation as we move forward. So one piece of this is to optimize a combination of the viral FCR vaccine antigens, looking at different combinations like GP34 plus GP68, and all 3, et cetera, to essentially maximize that Fc-mediated immune function. And then the other thing that we're looking to do is isolate viral FCR-specific monoclonal antibodies from our rabbits that will be important research tools for epitope mapping of those functional antibodies that'll help us understand what parts of the protein you really need to get good responses against to elicit this response that we're looking for. These monoclonal antibodies even have potential to be therapeutic agents themselves, which is something that we could look toward down the line as well. So in addition to that, we have some other future directions of this work that I'm really excited about, which is further optimizing the antigen combinations. So beyond just multiple viral C receptors, including other C and B antigens to help maximize that FC-mediated response. And then also evaluating efficacy. We can't do that in a rabbit model because C and Bs are highly species-specific, but we have recently identified BioLFCR homologs in rhesus CMV, which gives us an exciting opportunity to evaluate this vaccine concept in the rhesus macaque challenge model, which we have significant experience with in Dr. Permar's lab. So with that, I will acknowledge my wonderful mentor, Dr. Sally Permar, and our amazing lab group for all the work that they do, as well as our major collaborator on this project, Dr. Philip Kolb at University of Freiburg. And then, of course, our funding sources, specifically the Michelson Medical Research Foundation and Human Immunome Project for this funding. And I'd also like to thank all of you for joining for this panel and looking forward to discussion.
Wonderful. Thanks, Claire. I'd like to remind our audience, you can start submitting your questions for our panelists in the Q&A tab and voting on other people's questions you would like to hear answered. And now I'd like to introduce our third winner, Dr. Siyuan Ding. Dr. Ding is an assistant professor in the Department of Molecular Microbiology at Washington University in St. Louis. He's working to develop a dual vaccine against two of the leading causes of infectious diarrhea globally, which are rotavirus and enterotoxigenic E. coli. These are highly lethal in many regions of the world, causing over 300,000 deaths annually worldwide. Using reverse genetics, Dr. Ding has developed a live attenuated rotavirus vaccine that also encodes part of the ETEC toxin from E. coli. The oral vaccine passes to the gut, where Dr. Ding is examining if it induces localized protection and immune responses against both pathogens. His proposal is titled Immunogenicity and Protective Efficacy of Rotavirus ETEC Dual Vaccines. Thanks for joining us today.
My name is Siyuan Ding, and I'm an assistant professor in the Department of Molecular Microbiology at Washington University in St. Louis. My lab primarily studies the biology and pathogenesis of enteric viruses that infect the human gastrointestinal tract. Globally, diarrheal diseases are one of the major causes of morbidity and mortality among children under 5 years of age. In 2019 alone, more than 500,000 kids die from diarrheal illness. In the first year of life, the three leading causes of diarrhea are rotavirus, cryptosporidium, and enterotoxigenic E. coli. Currently, there are 2 rotavirus vaccines that are implemented globally, but no vaccines are available for crypto and ETEC. In this study, we propose to genetically engineer rotavirus to encode antigens made by ETEC. This is highly significant because if we can show the immunogenicity and protective efficacy of this dual vaccine, we can prevent 2 deadly infectious diseases with one single vaccine. I'm really humbled and honored to receive the highly prestigious Michelson Prizes Next Generation grants. It really supports a translational aspect of my lab that is distinct from all other projects.
Good morning, everyone. It's really a great pleasure to be here today virtually to present to you the work that's graciously funded by the Michelson Foundation. My name is Siyuan, and I am an assistant professor in the Department of Molecular Microbiology at Washington University in St. Louis. My lab has been working on rotavirus, which is a canonical enteric virus, and SARS-CoV-2, a non-canonical enteric virus, and how they interact with the host in the small intestine. So different aspects of mucosal immunology. And the work that I'm going to share with you today is some unpublished work from my lab using rotavirus as a vector to deliver exogenous antigens into the small intestine so we can use them to immunize children. So rotavirus is the leading cause of severe gastroenteritis and dehydration, diarrhea, vomiting, and fever in young children. What I'm showing you here is an epidemiology study that was done fairly recently, and it's surveying multiple sites around the world looking for the causes for diarrhea. And as you can see here, in the first year of life, rotavirus is the number one leading cause of diarrhea. It is also the number one leading cause of diarrhea in the second year of life, and number two from year 3 to 5. So even with the presence of several rotavirus vaccines that are fairly efficacious in the developed world, rotavirus is a major killer of young children, up to 200,000 deaths every year. So obviously we need to do a better job of reducing that number from rotavirus infection. But it also presents an opportunity because the rotavirus vaccines are readily available and given to kids around the world. So if we can manipulate the virus, there is a chance of using rotavirus to encode antigens to vaccinate against other enteric pathogens. And there is really a lack of such mucosal vaccination platforms that's super underdeveloped. So the challenge has been the rotavirus is a segmented RNA virus, and it's hard to manipulate. In 2017, a real game changer for many of us who study the biology of rotavirus infection came through. Takeshi Kobayashi in Japan developed this reverse genetics-based system to rescue recombinant rotaviruses from cell culture. So here, using two helper plasmid, one is a fast protein encoded by real virus. It's a fusogen that brings the cells together through syncytial formation, and also a vaccinia RNA capping enzyme that caps the 5' end of the mRNA to increase stability and translation. He co-transfected the 11 plasmids, encodes different genes of rotavirus into the same cell, and from there we can make recombinant viruses, in this case a simian monkey rotavirus strain that replicates almost at the comparable levels as the wild-type virus. So this is a great system because now we can actually manipulate the virus to use for mucosal vaccination purposes. But the problem has been that this S11 monkey virus replicates to high titers in vitro, but a lot of the viruses that we're working with, especially after expressing exogenous antigens, the replication is attenuated. So we need a better system to rescue viruses. So previously in my lab, we introduced three important changes that help us to increase the efficiency of this reverse genetic system. First, we used the MA104MV cell that stably expressed the V protein from parainfluenza virus 5, which degrades our STAT1. And we also expressed the BVDV N protein, which targets RF3 for degradation. So essentially, this MA104MV cell is a double knockout cell that lacks both RF3 and STAT1. And that seems to help because the viruses are susceptible to interferon. And by taking out the interferon system, we can recover more virus. And we also tripled the amount of the two plasmids that are used for rotavirus viroplasm formation. And thirdly, we replaced the vaccinia RNA capping enzyme with that of ASFV and fused with a T7 polymerase, so providing more T7 into the BHK T7 cells. And with that, we're able to rescue multiple viruses that are attenuated. But the systems always are great until they fall short. And as we're rescuing more attenuated viruses, we encounter more troubles. And so we decided to take a unbiased, agnostic approach. And in this data, unpublished data that I'm showing here, we took MA104 cells and we did a genome-wide CRISPR knockout library. And we infect them with a rotavirus-expressed GFP. And from there, we sort the super high GFP positive cells and the super low GFP cells. And the rationale is that by knocking out certain antiviral genes, the cells will become more susceptible to virus infection and thus producing more GFP. And from the screen, we identified two genes, SERPINB1 and TMEM236, that seems to support higher levels of rotavirus infection. And here, using rhesus rotavirus as an example, we can see 6-fold more virus production in these knockout cells. And we can see the same thing in RV5, which is the name for Rotatec, one of the rotavirus vaccines produced by Merck and used in the US and globally. And by combining the SERPINB1 knockout cells with a C3P3G3 capping enzyme, we're able to make a more efficient system that helps us to rescue multiple viruses, including especially the ones that we cannot rescue with the previous two systems. So it's all great, right? We have a great tool, we can rescue multiple viruses, but what can we really do with it? In a proof of principle study from my lab, we use rotavirus as a vehicle to encode the VP1 protein derived from human norovirus. Now, human norovirus won't able to infect mice, so this is only to show that this dual vector system is immunogenic. And by encoding the VP1 in the rotavirus genome, we're able to rescue this virus, and it has a modified gene that is way bigger than the original unmodified gene, as seen here by the RNA gel. We can show by Western blot that this one virus now can encode the VP1 protein. And when we stick it in mice, importantly, we can show that by both, I'm showing you here the serum IgG data, but we also have fecal IgG as well as fecal IgA data, that these mice mount a robust response to the dual vaccine. They can make antibodies that are neutralizing to both rotavirus and human norovirus. So this is all great, right? We have a system that we can use to rescue these viruses, and this virus seems to be immunogenic. The hurdle is that because murine, the mouse are not susceptible to human norovirus, we cannot really do a challenge study to show that the dual vaccine platform is protective. So we can show that it's immunogenic, but that's where we have to stop. So we're turning to different pathogens that we can use for the challenge system. And I picked two bacterial pathogens that are highlighted here. One is called enterotoxigenic E. coli, or abbreviated as ETEC, and the other is uropathogenic E. coli, abbreviated as UPEC. And I picked these two because several reasons. First, they are both important pathogens in the developing world as well as in the developed world. And second is they have very well-defined antigens that we can target. In the case of ETEC, we're targeting the heat labile toxin, which is composed of two subunits, A and B. And so in other words, this vector, using rotavirus to encode the ETEC vaccine, is not to prevent the ETEC from colonization. It's to prevent the toxin production and neutralize those toxins. So the bacteria, although they can adhere and they can make toxins, the pathogenesis is really neutralized. And for UPEC, we're targeting the FIMH, which is the tip of the fimbrion. That's these little sticks that sticks out, and FIMH is the tip of these fimbrion. So that would block the bacteria from adhering to epithelial cells. And the last reason is that I have really strong collaborators at WashU who can help us with these challenge models. With ETEC, we can just oral inoculate the virus in, and with UPEC, we have a catheter-induced infection model to mimic nosocomial infection in female mice. So what I'm showing you here is very preliminary data that's currently ongoing. Again, we can show in vitro that by the RNA gel, this modified gene is bigger because now it's expressing the beta subunit of the LT toxin from the ETEC. It's robustly produced in cell culture. The virus in vitro is really not attenuated after the expression of this large gene. And we also made different versions of this. We have one with just the LT alpha subunit that is contain the double mutant, so it's not really toxic by itself. We have another one that's just the beta subunit, which is shown here. We have one that encodes both, which is the DMLT. And then this is our inoculation scheme. So we have 5-day-old pups, which mimic the human babies. We give them one inoculation orally, and we measure virus shedding and diarrhea for 4 weeks. And then we follow up with a booster shot. And then we collect more stools, and at the end point, we collect the serum to measure the circulation IgG as well. So this is still work under progress, but comparing the parental virus, which is the rhesus rotavirus, with the ones that are encoding the UPEC FIMH, the ETEC DMLT, the LTB alone, and the FIMH plus LTB. So this is a combination of two rhesus rotaviruses encoding different antigens, because FIMH is known as a molecule targeting M cells in the gut, supposed to be boosting the mucosal immunology, and LTB can serve as an adjuvant by itself. Actually, in this case, in very preliminary analysis at week 4, in the feces, we're seeing more anti-FIMH IgA compared to the FIMH alone group, suggesting that the LTB, although we wanted to set out to study it as a target by itself, it seems to have some adjuvant efficiency and boost the FIMH response. The rotavirus IgA response, importantly, is not attenuated by the including of these exogenous proteins. So to summarize what I've shown you today, the rotavirus encoding the human norovirus VP1 is immunogenic in mice. Because we cannot proceed with the challenge model, we're moving to bacterial pathogens that are in the gut. And in very preliminary analysis, we're showing that the inclusion of LTB seems to boost the immune response against the other foreign antigens, such as FIMH. And importantly, when we're considering dual vaccines, we don't want the rotavirus antibody response to be attenuated by the inclusion of these antigens, and that doesn't seem to be the case. And I just want to highlight that mucosal vaccination platform are really urgently needed, because we see from the SARS-CoV-2 pandemic that the mRNA vaccine given parenterally is very effective at blocking severe diseases and death, but it's really not causing long-lasting mucosal IgA response, and that seems to be needed for the long-term immunity, especially in mucosal surfaces. And with that, I would like to thank the people in the lab who did the work, especially Yingxing, who did the CRISPR screen for MA104 cells, and Takahiro, who led the work with norovirus VP1 work, and Juhi, who is a PhD student in the lab, who is currently doing the collaboration on the bacterial toxins and antigens. And with that, I'm happy to take any questions and looking forward to more interactions with you guys during the social hours.
Okay, wonderful. So welcome all of our awardees here on screen. We'll now jump into the Q&A segment. So our audience, go ahead and input any questions you have for these panelists or for the collective group as a whole. And we will get to your questions. I'll start out with a few. Siyuan, you mentioned at the end of this need for mucosal immunity in many other contexts, including SARS-CoV-2. Can you expand on how your platform could be used more broadly and against which pathogens, and what kind of other vaccines you might expand to if this approach is successful?
Sure. So first of all, let me just say I'm super thankful to the Michelson Foundation and the Human Immunome Project for funding the more translational arm of my research that is riskier than some of the other projects going on in the lab. That is a great question. I think again, if the parenteral vaccine is great, it bought us time, right, and to prevent people from dying, but it really, at least for SARS-CoV-2, it's not blocking the transmission. So really need to get those mucosal IgA at the places to where they can block the transmission. So I think if you're looking at the large epidemiology studies survey around the world, at least for enteric viruses and pathogens, the leading causes are rotavirus, ETEC, Cryptosporidium, which is a eukaryotic parasite, Shigella, Vibrio, two more bacteria, and norovirus and adenovirus. So I think anything with a well-defined antigen may work. This idea of piggyback onto the rotavirus vaccination platforms.
Okay, great. Thanks so much.
Thanks.
And Dennis, for you, a similar question. Does your work have implications? Are the implications different for HIV vaccines versus influenza vaccines, COVID vaccines? And do you have to study each of those separately when it comes to antibody feedback? How do you collectively think about this in terms of vaccines as a whole versus individual applications thereof?
Right, that's a great question. I think in some ways, when someone talks about a feedback mechanism that seems to be kind of operating in the circuit way of how we think in a negative way, it's always people's alarm bells go off. But I think exactly how you pointed it out. While I do think you have to put it in two kind of categories. The mechanism, I think, like the principle, is probably the same. But as you completely correctly point out in your question, each case will be different. And I think that's definitely the case here, because the mechanism, the way it evolved, presumably evolved because it's evolutionary beneficial. And I think we see this quite well with SARS-CoV-2, where, multiple boosters or multiple infections just broaden your immune response, and that's a good thing. But and then I guess also the viruses that I mentioned, like HIV or influenza, there you really want to hone in on a target. And it seems that this is a mechanism that in a general way counters such a honing onto a single target. And in those cases, you really have the quote unquote negative implications of antibody feedback. So definitely very context dependent.
And so is that dependent on the pathogen itself or the vaccine, how the vaccine is designed as to if it's negative feedback?
Right. No, I think it's mostly the pathogen. I mean, I can't really comment on whether vaccine platforms have different, I guess, susceptibilities to these mechanisms. Probably, it has to do with antigen dose as well. But I think mostly it has to do with the target. If you only have one Achilles heel that you need to strike, then it's a problem potentially. On the other hand, if you have many targets, then it's not an issue at all. So I think mostly it's the pathogen, the antigen that matters.
Yep. Okay, great. And Claire, for your work, it's super fascinating that CMV has evolved this human-like Fc receptor protein to wield for its own benefit. Do you have any thoughts or comments on how that could have, how it's evolved to be like that? It seems so so clever of it to do so.
Yeah, so CMV and herpes viruses generally are very good at immune evasion. They have just massive genomes that encode all kinds of proteins that help the virus, balance sort of with the immune system, which is why they are able to, maintain infection over the lifespan of the host. But for the viral FCR specifically, it's really interesting because these proteins really are not related to the host Fc receptors, nor are they related to each other in terms of sequence homology and things like that, which kind of suggests that they evolved kind of independently. And it's interesting because there are other viral Fc receptors in some other herpes viruses like mouse CMV and HSV. But those actually have other functions besides the viral Fc receptor function, which I think is sort of leading other folks in the field to think that, they probably functioned for something else before and then have kind of over time evolved sort of a specialized function in the IgG binding, because we don't know, at least for the human CMV ones, nor the rhesus CMV ones, how they might function besides that, at least at this point.
Interesting. So then because they're not so directly related to the human ones, is this strategy of blocking them, is there concern that that would block the natural human Fc receptors, or would that be totally different?
Yeah, so that's always a question that we get is is there any concern of autoimmunity elicited by this kind of vaccine? And that's exactly why we think that it probably won't. But of course, we're going to check and make sure there aren't antibodies elicited against the host FCRs, just, as a final check.
Okay, great. So we've got some questions coming in from the audience, which is great. Go ahead and vote if you're watching on which questions you would like to hear answered. I'll start with a question from the audience for Dennis. And the question is, can you comment on the relevance of antibody feedback for older age groups? So does the antibody feedback happen more or less in those that are more elderly? Because we know they're responses to vaccines can be quite different than those who are younger.
That's a tough question because I'm operating in the absence of data here. So it's going to be a speculative answer. I think, once again, it may depend more on the actual antigen, on the pathogen, than on the host in this case. So when you're thinking about antibody feedback, you'll always have to consider the two arms of it. One is, what's your cellular repertoire, like your B cell repertoire, whether that's mostly coming from memory or mostly from naive cells. And, do you have a lot to offer? Or in the case of an older person, maybe do you have fewer to select from?
so more susceptible to antibody feedback. And then at the same time, what's happening in the serum. So what is the antibody repertoire in older individuals? And I think the latter we don't know about, because polyclonal antibody responses are often hard to disentangle. For the former, I think I'm going out here on a limb, but I think older individuals are known to have like reduced B cell repertoires. So it's possible that, they also accrued more antibodies over time. It's possible that the effects would be more pronounced, but this is pure speculation. And I think much more important than the question of old versus young is going to be the actual, antigen and how our human immune response kind of canonically responds to it. So that was my political answer that doesn't really answer the question. Yeah, was a good question.
I think that's a really great insight into what could happen. We don't exactly know, but good, interesting thoughts about B cell responses and things like that. And then for Siyuan and Claire, I think that also has relevance. Have you thought about how what you're doing might be different in how it elicits immune responses in more elderly age groups? I know yours are kind of targeted towards younger patients. So any thoughts or comments on that? Maybe Siyuan, if you want to go next.
Yeah, maybe I can start. So at least in the US, so the majority of the kids get rotavirus vaccine within the first 6 months of age. So there are two doses for Rotarix, which is the GSK vaccine. The kids get it at 2 and 4 months of age. And then for the Rotatech, which is the Merck vaccine, you get it 2, 4, and 6 months of age. And so we really get exposed to the antigen very early on. And then basically throughout the life, we're antigen experienced, right? So sometimes we refer to the globe as covered in the fecal veneer. Whether that's an accurate analogy or not, I don't know. But it's thicker at some places, but it's thinner at other places. But it's all covered in there. And my point is, we're always getting that exposure from rotaviruses, whether it's a human rotavirus or animal rotavirus. So by the time that we're adults and we're elderly, we're really antigen experienced. So unfortunately, this strategy won't work because we would already see the rotavirus already. And then if we give the virus orally, it will be counteracted by the host response very robustly. So again, we haven't done experiment just like Dennis. I'm just being very speculative here, but my guess would be that the host would mount such a quick recall response that the live virus won't able to have enough time to make the antigen for the other pathogens. So this will be a strategy for very young kids early on in life. Yeah.
Would there be other vectors that might be potential, or could you modify the rotavirus vector so that it would elicit a response?
So there's a lot of interest in engineering nanoparticles that can basically go through different route of administration. And I think if we can make a strong particle that does not look like it's something that the body has seen before, there is a potential of doing that and bring something into the gut. But the other enteric pathogens like the norovirus and astrovirus, we don't really have ways of manipulating those yet. So that's where I think the mucosal vaccination platform, at least for the GI tract, is falling short.
Interesting. And Claire?
Yeah, so kind of similarly, like CMV is a ubiquitous virus that most people have been exposed to, by the time they reach adulthood. And, most people are infected by the time they're like 40 years old. So this, I guess, like a vaccine for CMV is really intended to protect like very vulnerable populations, specifically like the newborn for mother to child transmission of CMV, but also like severely immunocompromised patient populations, like people undergoing transplant is another population that's severely affected by CMV. And so I think, like, we're really trying to get people or like get immunizations to people when their immune system is strong and able to make that immune response. And, hopefully that will be durable and protect against complications that can result from reactivation or a superinfection. And I think sort of at least like in the context of our research, we're really focused on the congenital context. And so our target population is really moms, ideally prior to getting pregnant, but potentially even during pregnancy. And there's also suggestion that like toddlers, the under 5 age group is like sort of super spreaders of CMV. And so there's some work going on looking at that as a potential target population for prevention of congenital CMV, even just to prevent infection of moms who, have a toddler at home or are exposed to kids that age.
Yeah, great. I have another question for Siyuan. You had mentioned many different pathogens that the approach could be used against. Have you thought about, or is there potential to create more than a dual vaccine? So incorporating a number of different, is there a limit to how many you could vaccinate against at once? And yeah,
absolutely. So again, following up on what I mentioned, so there are 2 vaccines that are used in the U.S. One is the Rotavirix, the GSK vaccine. It's a single virus strain isolated from a human kid, I think from Philadelphia in the 1988 or 89. And there was seropassage in cell culture to cause this attenuation. So it's a live attenuated vaccine. And then the Merck vaccine is actually a combination of 5 different viruses. So these are essentially bovine. So they're cow rotaviruses that encode the human rotavirus spike proteins on the out capsule. So they look like a human virus from the outside when the immune system sees it, but it's inside it's actually a bovine rotavirus, which replicates very low level in human baby intestine. So it basically does not cause diarrhea, and it induces protective immunity. So for the Merck vaccine, which is the pentavalent one, there is actually a chance of modifying each one of them to encode against multiple pathogens, and that's definitely of great interest. Yeah.
When you do that, is there a concern about it being too reactive with the immune system just kind of freak out that there's so many things can it be hyper immunogenic? Is it problematic at all?
Yeah, so that's another great question. I think for any manipulation, there needs to be safety trials very early on and large scale studies. So again, the 2 vaccines, the Merck and GSK vaccine, were rolled out in 2006 and 2008. And actually, prior to that, there is another rotavirus vaccine called RotaShield that was introduced in 1998, approved by FDA, and went on the market, actually used in a lot of kids, and it was withdrawn after 6 months later. So the problem seems to be this thing called interception, which is basically the gut telescoping, like the lower part goes on top of the previous part, and then this part is basically lacking the blood. And you can do a simple procedure called ballooning, just blow it up. But a lot of places, they actually go to surgical removal of that section. And it's not a great risk. I would say it's like probably 0.1% increase, which is why they missed it in the very early on trials. But when it was actually used in the market, thousands of kids, hundreds of thousands of kids got it. And that's when they noticed it and they withdrew it. So for every single vaccine, we want to be super careful, right? Because, again, we're all pro-vaccine, but we want to really make sure it's safe and to use, especially in babies.
Mm-hmm. Great. Claire, we've got some questions for you from the audience. One question, have you considered RL13 as an FC receptor? There is one paper suggesting it binds to FC, but it's highly polymorphic. And how polymorphic are the 3 FCRs you've been studying?
Yeah, so the gene product of RL13, at least like in the hands of our collaborators that we're working with, has not been easy to express, in part because it's just not as present. There's really high turnover of that protein from the cell membrane, so it doesn't function as well as a viral FC receptor as the others. And so particularly given the difficulties in producing that protein, we have decided to focus more on the ones that are easier targets. And yeah, in terms of polymorphism, it's quite interesting because GP95, which is encoded by RL12, is like one of the most polymorphic regions of the CMV genome. And so we're not sure that that's going to make a great vaccine target, but preliminary results from some of the things that I'm working on now shows actually pretty good breadth across some CMV strains that we have in the lab in terms of that ADCP function that I showed in the presentation. And the other two, there's some polymorphism in GP68, which is like a splice variant of two genes attached together. And then RL11, which encodes GP34, is pretty conserved. There's really only small changes across virus strains.
Okay, great. And Dennis, for you, I'm wondering, there's been a lot of efforts to try to make some kind of a pan-influenza vaccine. Does your work have any implications for how to broaden the antibody kind of production from how a vaccine should be designed to enable a broader antibody production? And does antibody feedback come into that at all?
Yeah, it's a kind of a key question, and ultimately, I think we and others think, and like there's been studies out also on the concept of another immunological old concept called original antigenic sin, and I think there's a converging kind of like idea in the field that this may be at least in part due to antibodies, pre-existing antibodies, competing with certain naive cells where they have an easier time to reactivate, in a re-encounter, and that's why you keep this essentially response to that first insult, if you will, of or first variant, at least on the plasma level. So we do think that antibody feedback as a mechanism would at least partially explain this phenomenon of kind of antigenic seniority. Now, the other part of your question, which is how to circumvent this, I'm going to be very honest, like, I don't know. I think it's very hard, and maybe this is not the most optimistic view, but I think if antibody feedback operates the way we think, then it seems to be a very hardwired system of immune diversification. So really, like, making a vaccine for influenza, let's say, or a vaccination series where you have selective exposure of the stalk, which is where you want your antibodies to essentially bind, which is like not the top of the viral antigen, but it's kind of on the side, which is hard to get to in the beginning anyways. To have that response be made, but not produce antibodies against it that will then subsequently feedback against similar types in the next round, that's a tall order. So I think for the time being, we have to be realistic and just say that, our kind of repeated annual vaccinations with kind of the best we got, which is not a very good vaccine, but it's still, fantastically better than not being vaccinated.
Something.
That's going to be for the next couple of years at least. I don't have a direct kind of like segue into like how do we circumvent this. I think it's very hardwired.
And any other insights into how we should be maybe designing boosters differently for other types of infections, whether it's viral or bacterial?
Right. Yeah. Well, I think I don't think so. I don't think we should start redesigning everything, because ultimately, when you think about something conceptually, it's not necessarily the same as like being pragmatic. And I think I showed this in one of my slides that one of the reasons for boosting is like not this much taller order of trying to elicit broadly neutralizing antibodies, but it's in fact just to like bump up the antibody levels in your blood against, the oncoming cold season, influenza, whatever it may be. And I think boosting, for all we know, does fulfill that function quite well. We all kind of, hopefully get our like annual shots. And ultimately, even if this is what I call subpar, due to antibody feedback response, it's still going to be more than before we boosted. And hopefully that's going to protect us. So I don't think I think it's something to keep in mind for future vaccine development, like as a mechanism and something to consider, which hasn't been considered in the past necessarily. But I think clinically right now, I wouldn't jump to any conclusions, and I would just, go with the things that are, tried and tested until we have a better understanding.
Okay. Do you guys have any questions for each other? I know you kind of do somewhat related, but somewhat different work, but did anything spark any ideas or questions when you were listening to each other's presentations?
Yeah, maybe I can go ahead. I have a question for Siyuan, which is, you explained that one of your use cases is the piggybacking of the norovirus VP protein. And so it kind of goes to a question that Shannon asked earlier a little bit, is like, we know that norovirus, the immunity, one of the issues is, the heterogeneity, the genetic diversity of that virus, and how fast it, is very different from, one strain to another. I'm not talking now about putting two different antigens altogether on there, but what do you think? Is it a good platform, the one you're developing, to just encode for like a breadth of, like, let's say 100 different strains of it that you could just co-transfect? What are your thoughts on that?
Yeah, no, that's a great question. So I think we picked VP1 because it kind of self-assembles, right? So when we express a ton of it, it just makes this virus-like particles, which is great at inducing immunity. There are specific smaller fractions of the VP1, which is called the protruding domain, the P particles, which is an even smaller fragment. So I think the current limitation is the capacity of how much we can squeeze in to the rotavirus genome, right? So it's not like you can make it's a sphere, right? So it has some restrictions for how much you can pack inside the genome. So I think for VP1, we may be able to pack only 2. But if we use the smaller fraction, which is the P, we may be able to put multiple genotypes in there from the human norovirus to kind of cover the major serogroups that are circulating around the world. Yeah.
Very cool.
Thanks. Yeah, and I can, I guess, chime in on that pan influenza vaccine a little bit. There is also a lot of interest in designing the pan sarbecovirus for coronaviruses, just for pandemic preparedness, right? Because we've seen that antigenically, the current Omicron variant is actually more distant, or more distant from SARS-CoV-2, the ancestral strain, compared to SARS-CoV-1, right? So I think there is a lot of interest in looking at the spike, and then because there are a lot of viruses that we haven't surveyed, we haven't identified yet in bats and pangolins, and those could cause the next pandemic. And what we really need is something that can have that breadth to cover a lot of them, even the ones we haven't seen before, right? And there is, I think, NIH is putting a lot of money in terms of designing these pan-coronavirus vaccines, whether it's a merbeco or sarbecovirus, so that it will buy us some time. And when we get it, it will, again, won't necessarily prevent transmission, but prevent the severe diseases deep in the lung. But that can buy us some time to design the specific ones that have better efficacy.
Great. Any other questions for each other?
Sure. I had another question for you, Dr. Ding, about like the efficacy of the rotavirus vaccines in like low and middle income countries. I know that's like an issue where the efficacy tends to be lower. Do you anticipate that would be a similar problem with these dual vaccines, or would you hope that would somehow help address this problem?
Yeah, no, that's a great question. And I think I'm with Dennis on that. Having something is better than nothing, right? So just to give the audience a little bit of context, so the current vaccines, the Merck one, the GSK one, they're like 98% effective in the developed world, right? And the same vaccines, it's probably multigenic, the underlying mechanism, but when they're given to kids that are in Africa or India, Bangladesh, the efficacy drops to suboptimal levels, and we're talking about like 40%, 50%, right? And that's why we have still so many kids dying from rotavirus infections or so early in life. And I think having the dual vaccine won't fix that particular problem, but it's going to fix the other problems that hopefully, because ETAC and cryptosporidium and other Shigella and Vibrio are still major killers in many parts of the world, and they actually share this overlapping epidemiology. So a lot of the places where rotavirus are killing the kids, the same, the bacterial pathogens are doing the same thing. So it won't necessarily improve the rotavirus vaccine efficacy just on its own, but it will help to prevent the other ones. And again, I think having something is better than zero. And with we've seen really with the rollout of the rotavirus vaccine since 2006, the number has gone down from 500,000 kids death every year to 200,000 death every year, and that's a big improvement.
Great. We've got a few more questions for Claire from the audience here. Someone comments that CMV infection upregulates host FC receptors. I guess there was a famous Nature paper years ago suggesting that this might provide a pathway for HIV entry into non-CD4 cells and HIV susceptibility. Do you have any comments on that activity and how that might come into play with what you're doing?
Yeah, I can't say that I have done a ton of work or reading on like HIV CMV co-infection, although that I know that is a popular group to target to study congenital CMV infection, because mothers who are also living with HIV are more likely to transmit to their baby because they have lower CD4 counts. But in terms of the the upregulation in FC receptors, I don't know a ton about that, so I can't really speak to it.
If they're HIV positive, you said they often transmit more to the fetus. Do you know how they would respond to the vaccine? Would they respond less to the vaccine as well?
Yeah, so we have done some research in the rhesus macaque model, investigating like CD4 T cells as like really using that as a model system, like depleting the CD4 cells out of the pregnant rhesus macaque and then infecting them with CMV. And we get really consistent vertical CMV transmission in that model. And we're still we're doing work now to really parse out exactly why that's happening, because CD4 cells have such broad functions across the immune system. But we have seen in that model system that the antibody responses are not as affected as we would have necessarily thought with lacking CD4 cells. There's really just a delay in the response, but it's not the magnitude of the response has not been affected. So I think for a vaccine that is really targeting eliciting antibodies, it's potentially less of an issue in that population.
Okay. And then there's another question that is the neonatal FC receptor blocked by the CMV ligands, and does this impair transplacental transfer of IgG to the fetus, both in terms of natural CMV infection, but also in terms of what you're trying to do with your vaccination?
Yeah, for sure. I think I remember seeing a paper at one point describing a mechanism that CMV downregulates the neonatal FC receptor. And there's certainly implications for that in terms of placental transfer of IgG. But in terms of the viral FC receptors and how that may play a role, I'm not sure that has really been looked at. But I think that definitely could be an interesting question. And there are also other, functions of FC in the immune system. We really haven't looked too much into complement and how that's affected by the viral FC receptors, nor the intracellular FC receptors like TRIM21. So I think there's still a lot more, basic science even to do in this space, which is really interesting and exciting.
Great. So we're nearing the end of our time here. So my final question to all of you is, what aspects of your work were hard to get funded through traditional mechanisms like NIH grants and things that these Michelson Next Generation Grant Prizes will enable you to do?
Maybe I can start. I think with the current NIH funding climate, it's really challenging because Dennis and Claire can chime in, but I think most of us will apply to NIAID, which is the big NIH branch for infection and immunity. And their current funding line for R01, which is the standard big grants, is at 10%. So basically, for every 10 grants, really good grants going in, only one of them will get funded, right? And that's been super challenging for a lot of people, and especially with the line of work that we're doing. So I think this really provides a lot of opportunities for my lab and to sustain the kind of work that we're doing. It's been a huge help. Yeah.
Great. Dennis, maybe you go next.
Yeah, so actually in my case, first of all, what Ron said was definitely, is definitely an issue. NIAID is a tough place these days. But in addition to just, the pay lines and getting through the cut, I guess, it's also that it's hard to pivot quickly, right? I mean, most NIH grants have a long time horizon. So there needs to be a long plan. And if you're pivoting your research quickly, or if you essentially would like to, propose a maybe like somewhat out of the box concept, which I think we all do, in a way that, would give funding maybe only for a year, and also raise exposure and can pretty much be also like a bridge to other funding based on that exposure. I think that's where the Michelson Award for me stands out, because it is so, like you apply for it. The cycle is in 2 phases, which is also, I think, nice. You don't have to put in a whole application right away. I think that's also a reason why I would encourage people to apply, because you're going to know fairly early whether you should, in fact, because, writing grants is it's not easy. It takes time. Whether you have to put in a whole application package. So I think for me, it's both the modular design of the application and also the fact that, it's more of a short-term funding horizon that made it particularly attractive in a way that maybe NIH, does not offer.
Mm-hmm.
Yeah, and I think just to add on to what has already been said, one of the nice things about this grant for me is that, as a postdoc, I cannot apply for an R01 or NIH mechanisms as a PI, because I'm not at that level yet. And so the Michelson Award is nice because I am listed as the PI of this grant and have, an additional track record of funding that is recognized, whereas other grants that we've applied for, I've had to contribute to things under my PI's name, which is great, and I'm happy to do that because we're getting funding for my work. But at the same time, this is a really exciting opportunity to further my career based on that.
Great. And anything else you wanted to add? Felt you didn't get to say yet? Anything before we wrap up?
It was amazing to see the co-recipients in person and hear about their work, and thank you and the organizers just for organizing this event. That's all.
I really thank you guys for putting this together. Yeah, it's great.
Our pleasure. It's been a really great time talking with you all and exploring your really interesting work, and we really look forward to seeing what comes out of it. So unfortunately, that's all we have time for today. And yeah, thank you so much for coming to discuss the future of science and immunology with us, and for our audience for attending and contributing questions. The ePanel will be available on demand, and we will email the audience when the recording is available. We invite everyone to now join us in the networking lounge to ask the speakers any remaining questions you might have one-on-one or small group conversations, and to connect with other audience members to continue these conversations. The Remo platform allows you to jump between small group discussions as you would during a coffee break or cocktail hour, and you can just click on the button to join on the homepage or in the closing screen once we conclude. For our audience, the 2024 Michelson Prizes Next Generation Grant Application Portal is now open for those aspiring immunologists and vaccinologists out there to apply for next year's funding, so that's very exciting. The portal closes on June 9th, so get those applications in soon, and you can find out more about that through the event information tab on the left side of this page. And finally, we will be hosting an in-person award ceremony for a subset of the Michelson awardees during our upcoming conference on positive strand RNA viruses, interdisciplinary advances in virology, pathogenesis, immunology, and technology development, which will be held in Killarney, Ireland in October. So you are all welcome to join us there. You can apply for a scholarship to attend or submit your abstract by July 23rd for the chance to be chosen for a short talk. And don't miss the early registration deadline of September 4th for a $200 discount. You can also find more information about that in the event information tab on the left or on the Keystone Symposia conference homepage. Thanks again for attending today, and we hope to see you in the networking lounge.